Hello and welcome back. We are here to do another little mini lesson on human genetics in chapter 16 topics. This lesson is going to cover some chromosomal abnormalities. So what are some things that happen when either your chromosome number is incorrect or maybe the chromosomes themselves have been damaged? So prior to this, you should have a pretty good understanding of meiosis, knowing how the chromosomes split and divide in the process of gamete production, going from a diploid parent cell to a haploid daughter cell. And then also some of the inheritance patterns, dominant recessive, sex-linked traits. Those are some prior knowledge that you can be bringing into this particular video to help you understand the human side to chromosomal abnormalities. The first thing we're going to talk about is what's called nondisjunction. So nondisjunction is an event that happens in mitosis or meiosis. Here they're showing it in meiosis, but it could happen in mitosis. But the bigger problem is when you have nondisjunction in meiosis, because it's going to lead to an abnormal number of chromosomes in your gametes. And if that gamete is used to get fertilized, either the egg or the sperm, then the individual that was made by those incorrect gametes will have all of their chromosome number be mistaken. So here on this particular picture, they're just showing and tracking the X and Y chromosomes, but this could be true for any chromosome. It'd be really hard to put all 46 and then 23 chromosomes in these little pictures, but you could track this down for chromosome 1 or 5 or 15 or 18 or 21. So they're just using the XY as an example, but it could be any homologous pair. So nondisjunction, kind of the big picture idea is that during anaphase, when the chromatids or the homologous chromosomes are supposed to be pulled apart, they don't. And in meiosis, we have two anaphases. So there's two opportunities for this nondisjunction to take place. So we're going to do the top box is showing what happens when there's nondisjunction in meiosis 1 during anaphase 1. The bottom box is going to show you nondisjunction at anaphase 2. So if we're going to follow spermatogenesis, which is a production of sperm, which is what this picture is indicating here, we have our meiosis 1. Okay, metaphase 1 of meiosis 1, sorry, where your homologs are lined up on the metaphase plate. And then normally what's supposed to happen is they're supposed to separate. That's a law of segregation. Your homologs get pulled apart. So each daughter cell has a full complement of the chromosomes that they're needing. In nondisjunction, as you can see here, in this problem of anaphase, is that instead of the homologs being pulled apart, both homologs go to one side, and that's going to go into the product of the end of meiosis 1. So this is telophase 1. And eventually you would go through the second round of meiosis, and you'd get to telophase 2. And what you'd end up with is your 4 haploid gametes, but instead of having one sex chromosome in each sperm, you have 2. So this is a +1. It's got 1 extra chromosome in each sperm, and these ones are -1. They're missing a sex chromosome. They're going to have all of the other. If there's no problems with all of the other chromosomes, they'd have chromosomes number 1 through 22 perfectly fine. But these sperm, they'd have 24 chromosomes in those top 2, and then 22 chromosomes in the bottom 2. So if those were sperm that were used to fertilize an egg, there would be an unnormal, is that a word, unnormal? A not normal number of chromosomes in that fertilized egg. So if here's the egg over here, and it had its 23 complement of 23 chromosomes because it was produced properly, and this sperm went over here and fertilized that egg, what you'd get would be a zygote with 47 chromosomes, 1 additional sex chromosome. So if mom is carrying an X, this individual would have the X from mom and the X and Y coming from that non-disjunctioned sperm. If it was one of these other ones down here on the bottom, if one of these sperm came and fertilized that egg, they would have 22 chromosomes, and so then you'd end up with 45 chromosomes, the X chromosome coming from the mom, and no sex chromosome coming from the dad's sperm. So if either of those possible sperm, the plus ones or the minus ones were to actually get to fertilize the egg, you're going to have a problem with the chromosome number. And so this is what we call polyploidy, too many. Polyploidy is you have too many, like diploid and haploid. Diploid is 2, haploid is 1 set. Poly is more than your normal number, and aneuploidy is missing. You're missing some. Now, you can get this non-disjunction happening at meiosis 1. You can also get it happening in meiosis 2. So here is our metaphase 1. Here's our anaphase 1. Here's our telophase 1. Everything's good. Oops, that's not the right one. This is our telophase 1. But then in anaphase 2, take a look at what happens. So on that X chromosome, on the purple chromosome on the top there, instead of the chromatids pulling apart, both of the chromatids went to one side. And what that gave you is 2 sperm that are normal. So these are the normal ones here. They only have 1 of the sex chromosomes, which is what they're supposed to have. And here you have one that has 1 too few, and here you have one that has 1 too many. Or the other option is if you had non-disjunction in the Y chromosome, you would get the same problem, but with the Y chromosome. So if these problematic sperm were to go and fertilize a normal egg, you'd also get that aneuploidy or polyploidy outcome. Now, when this happens, what happens to that fertilized egg, the zygote, the embryo, the baby, the person? Well, there's a couple different examples of aneuploidy and polyploidy that we're going to take a look at. So probably the most common viable polyploidy is a trisomy, where you have 3 sets of chromosomes of a particular chromosome. And this first box here, this is trisomy 21, which is usually what we recognize as Down syndrome. So here you can see circled by that blue circle, for whatever reason, there was non-disjunction in either the egg or the sperm. You wouldn't be able to tell necessarily by just looking at the karyotype, but either the egg or the sperm had 2 chromosome 21s because of non-disjunction, fertilized with either the egg or the sperm that had their normal 1, and you ended up with 3 chromosome 21s. And then you get all of the phenotypic expression of Down syndrome. So this is also called trisomy 21. This one down here on the bottom, this is called Klinefelter's syndrome. So it would be like if we had one of those double X, either egg or in sperm on the last slide. And a sperm had non-disjunction and carried 2 X's, or the egg had 2 X's and it was fertilized by the sperm with a Y. Either way, you get 2 X's and a Y. Now, because this is affecting the sex chromosome, it affects some of the physical phenotypic traits of an individual's sex and kind of gender characteristics. So normally, an XX person is a female, and an XY person is a male. So what happens when we have XXY, when you have kind of both combinations? Well, in this particular case, individuals with Klinefelters, they are male because they have the Y. The Y kind of confers maleness. But because there's an extra X chromosome, they take on physical appearances of some female characteristics. So you can see things like breast development, female pattern, pubic hair, kind of wider hips, narrower shoulders, poor beard growth, fewer chest hairs. So all of these are kind of tendencies more towards a feminine body style, but they do have male reproductive parts and they are technically male. They just have an extra X chromosome. In this karyotype over here, I don't have a picture for it because these individuals, there's non-disjunction, but it is X, Y, Y. So it's trisomy. There's 3 sex chromosomes. These individuals are male because they have the XY, they just have the extra Y chromosome. And phenotypically, they're not really that different. They can't be visually differentiated between other males. So there's not as negative outcomes, if you will, for individuals who are XYY. Okay, so those are some examples of trisomy in the human condition. What happens if there's monosomy, if you only get one particular chromosome? Now, if you get monosomy in any of the other chromosomes, 1 through 22, usually it's a natural miscarriage that the organism would not be able to survive. However, if you get monosomy with the X chromosome, where you have one X chromosome, they're female, but there are some phenotypic traits and some physiological changes and challenges that these individuals have. So you can kind of read through that list, and there's a picture. I don't watch her show, but I think she's part of like a crime show or a cop show or something. I know she's a TV actress. You cannot survive if it was monosomy with just the Y chromosome. That would also lead to a natural miscarriage. The embryo would not be viable. But monosomy, this is called Turner syndrome. So Turner syndrome is a viable monosomy of just the X chromosome. All right, so those are some chromosomal abnormalities when the number of chromosomes are incorrect, and that's usually due to nondisjunction of one of the steps of meiosis, either at anaphase 1 or anaphase 2. Some other abnormalities of chromosomes are their structure themselves, not just the number of chromosomes, but in the process of DNA replication in that S phase of interphase, there might be some problems that are happening. So this is just a list of some chromosomal problems that could lead to phenotypic and physiological diseases. So say your DNA is going through DNA replication, and for whatever reason, a certain section just kind of gets duplicated. So in this diagram, a section coded A, you might end up with 4 times as much of that A information on your chromosome. And so that's duplication. Another example is inversion. So as the DNA is replicating or maybe during crossing over, the traits, the loci, right, the gene location gets inverted and rearranged. Sometimes you might get a deletion where part of the chromosome just is lost somewhere. Again, that could be happening during recombination in prophase 1 or DNA replication. There was an error and just a chunk of the chromosome is missing. And then this last one is called reciprocal translocation, and it looks a lot like recombination, like what happens in prophase 1 of meiosis. But remember, that swapping of genes was on homologous chromatids. They carried the same information. So all they were doing was exchanging the allele versions of the same genes. If you have translocation between non-homologous chromosomes, you're messing up the pattern of how the information is supposed to be carried on those particular chromosomes. So there's some, there's probably lots of congenital and genetic diseases or conditions that can be linked to these abnormalities of chromosome structure. Number one, Fragile X. No, these are two. Sorry. These are two of those chromosomal abnormalities. So the first one is called 5p minus syndrome, also called Creux-de-Shaw, which is the P is is the short arm of the chromosome and the Q is the long arm. So it's kind of helping us map our locations. So in a 5p minus, the whole short arm of the 5th chromosome is missing. And what that leads to, so if somebody is diploid, right, so all their cells are diploid, they would have one copy of a normal number 5 and this shortened deleted version of the short arm, the P arm of the other number 5, and that leads to mental retardation. It's one of the most common forms of mental retardation in the United States. So that's called Creux-de-Shaw, which means cry of the cat, and it was named that because the infants who have this abnormality, when they cry as infants, kind of sound like a cat's wailing. The other chromosomal abnormality phenotype is what's called Fragile X. Okay, so Fragile X syndrome. Oops. So Fragile X is the product of duplication. So here we have our normal on the X chromosome. We have a short little repeat of C's and G's, so cytosine and guanosine repeats, so about 50 repeats, and that would be normal for the X chromosome on that particular area. But in Fragile X, what happens is that CGG repeats 200 to 1,000 times more than it's supposed to, and it makes kind of like a little constriction and it makes that X chromosome fragile. And what happens is you're going to get defective X chromosomes, which is inherited. It's almost like a sex-linked trait because it is inherited going along with the X chromosome. So you can kind of see this little distribution of not affected male mating with a carrier female, that's the yellow. So then you would get 50% of your offspring could be potentially normal, 25% would be an affected male, and 25% would be a carrier female. So Fragile X syndrome is the result of that addition, the repetition of a particular sequence. All right, so those are the examples of some chromosomal abnormalities I wanted to share with you. So nondisjunction leading to polyploidy or aneuploidy. And the chromosomal abnormalities, those different characteristics, so repetitions or deletions or translocations or kind of the swapping. So those are some chromosomal abnormalities that we can find in humans. All right, I will see you next time.